thing we've got left, actually, in terms of moving forward at the moment. Okay, so if there's no more pressing things, obviously, the chat is still open, then we are just a couple of minutes ahead of schedule, but that's great. So we move on to the second grand challenge, which is evolution. And to present and to discuss this, we have two people from the Czech Republic, Vladimir Hampl and Michael Hammond. So whichever one of you is taking it away, please do so. Thank you.
Thanks, Mark. Yeah, so I'll start, and then sooner or later I'll hand it over to Vlada. So on our first slide, we have the current phylogeny of euglenids with Phototrophs highlighted in green, Osmotrophs in blue, and Phagotrophs in black. At present, the photosynthetic euglenids have received the lion's share of attention and research, and obviously it seems that these phototrophs appear to have quite complex and unwieldy genomes. One of the points that we will be highlighting today is the importance of expanding our focus beyond this group to other representatives in our search for an easily resolvable genome. But we will start with our model organism of Euglena gracilis and the evolutionary information that we can gain outside of a well-resolved genome. So going to the next slide, our first evolutionary goal is to investigate how the endosymbiotic organelles, the plastid and mitochondrion, have been affected by the acquisition and the selective loss of photosynthesis. So if you look at this graph here, you'll see that in plants, efforts to resolve the mitoproteomes of organisms has been hampered, presumably from the small size of mitochondria and the hypothesized low abundance of proteins. And in protists, this is also hampered just from a lack of research focus. But this isn't so much of a problem in Euglena. So from profiling both the mitochondrion and the plastid, we have come to a group of sequences, 185, which are candidates for dual targeting. And this is really exciting because the prediction of dual targeted sequences is exceedingly difficult in silico. From a functional perspective, we know that the proteins that are most likely to be dual targeted to organelles include nucleotide metabolism, DNA replication, and tRNA biogenesis. And you can see that these are represented somewhat in our functional annotation. But more importantly, we have the opportunity here to profile these amino acid sequences and determine the criteria for dual targeting in euglenids. So is it that some of these proteins have an N-terminal target peptide to one organelle and a C-terminal peptide to another? Or do they possess ambiguous target signals that are recognized by multiple receptors of different organelles? So going to our next slide, as I highlighted yesterday, what's really key here is expanded organelle proteomics in different euglenid species to understand organelle interaction. So I would suggest here that Euglena longa is perhaps our best candidate given its previous analysis and the availability of its transcription. So as you know, Euglena longa is a close relative to Euglena gracilis and is distinguished by a non-photosynthetic plastid that is nonetheless essential for viability. So I'll just like to segue briefly to the Stramenophile supergroup. Like euglenids, this includes lineages that have acquired a photosynthetic endosymbiont, although in this case it's a red algal ancestor, and some lineages have lost photosynthesis secondarily, and there are lineages that have never gained photosynthesis. So they're like euglenids in this respect. A way in that they're not is they have well-resolved genomes and transcriptomes that have enabled researchers to make well-supported predictions in silico. So concerning organelle complexity, one of the interesting trends that they predict is that in species which have lost photosynthesis, the complexity decreases in plastids, as you would expect, but this is also shared in mitochondria. And this is similarly seen in species, the mitochondria of species that were once photosynthetic compared to species and close relatives that were never photosynthetic. You also see a loss in complexity. And so preliminary comparison of the transcriptomes of Euglena gracilis and longa against other mitoproteomes suggests a somewhat reduced cohort of ortholog groups in Euglena longa. So I think we have an opportunity here with these euglenids to verify these proposed trends with our experimentally available representatives. So I will now hand over to Vlada, who will discuss genome mosaicism.
Thank you very much, Michael. So I will continue with the second challenge which we decided to discuss, and that's the topic which has been already mentioned, the known genome mosaicism in Euglenida. And it's been known for Euglena gracilis that some pathways are in mosaic, and let me show you some data in which we try to understand the mosaicism in more detail by subjecting all predicted proteins of Euglena gracilis, the phototroph, and heterotrophic Rhabdomonas costata to phylogenetic analysis to understand the affiliation of these genes. And as you can see, slightly more than 3,000 of genes we were able to affiliate with some other groups. And most of them were related to excavata, which is kind of expected. A subgroup of Discoba and Kinetoplasta columns are subgroup of the first. So that's also understandable. And then, however, then we see a bunch of other affiliations to algae and also non-algal groups. Some of them are kind of unexpected. They might be correct, but we may also consider them as showing us the error rate of the method, something that we can call a gray zone of the identifications that we may but may not believe, out of which some of the groups are sticking. And as you can see in Euglena gracilis, those are viridiplantae, which is easily explainable by the origin of the green plastid. So these genes probably arrived to the nucleus of Euglena gracilis by endosymbiotic gene transfer. But what was unexpected, but already in a literature mentioned several times, also haptophytes and ochrophytes and maybe cryptophytes contributed to Euglena gracilis genome significantly. In Rhabdomonas costata, we don't see such contribution besides this column of viridiplantae genes, which may mean who knows what, but generally we think it's in the line of, it corresponds to the notion that Rhabdomonas has never had plastid in the photos in its history. And so one of the challenges we see is if we could somehow separate and pinpoint the provenance of the other than green algal contributors to the Euglena gracilis genome. And in general, I think we should study more genome mosaicism in more euglenids, like, for example, this very interesting Rapaza viridis, which I will talk a bit later about. The first, the third challenge that I see is to understand the plastid genome evolution, which, as you know, euglenophytes inherited the plastid genome from green algae, and the plastid genome originally, as nicely depicted in this figure from Ania Kankowska's study, contained inverted repeats and one type 2 intron. However, this structure has been quite turbulently changed during the evolution of euglenophytes. Some of them still preserved the inverted repeats, some of them lost them, some of them have tandem repeats, and also the introns became kind of unleashed. And in some lineages, and especially in the best studied Euglena gracilis, reached considerable numbers. But it is basically we have no idea what both the structure of the genome and the introns in the genome have what's the biological relevance of these, if any. The fourth challenge we decided to speak about is to understand the plastid biogenesis. It has been shown in the past by beautiful studies, for example, by Slavikova and et al. In Professor Schwarzbach lab, that the free membranes of Euglena plastid are the first membrane is crossed. The protein crosses the first membrane through vesicles, fusing with the membrane that are derived from ER and go through Golgi, which is not drawn here. And then potentially the protein enters the stroma via TOC and TIC complex. And we started with our proteomic data to put the proteins to the picture and identified some of them which might tether and help with the fusion of the vesicles, and we start to see the very modified and ruminant TIC complex. But how the proteins cross the middle membrane, it's kind of mystery because we don't see any TOC complex. But maybe but that's a wild hypothesis. There are present Derlin pseudoproteases similarly, like analogously to the plastids of red algal origin. And I think we would also need, should understand the origin of the membranes. The number of free membranes is kind of unexpected for secondary symbiosis, as has been pointed out by Professor Schwarzbach already. And we try to understand from where the membranes are derived by analysis of the lipids. We purified the envelope fraction of that plastid, and we have learned that more than half of the lipids are galactolipids, which are typical for chloroplast membranes. And from this, we derive that definitely more than one of these membranes has to contain the galactolipids, if half of them are in the fraction. Actually, the 53% is 2/3 of 80, which is a thylakoid galactolipid content. And so we think that 2 of these membranes are derived from the chloroplast, while the outermost is eukaryotic. And we also can learn more now with the proteomic data in which we establish the content of the Euglena chloroplast about the nature of the targeting peptide, which was shown to import the proteins into P chloroplast, for example. And so we can learn which amino acids are enriched and which not. However, in the context of the translocation machinery, it is, I think, quite interesting question. And I will move forward to the fifth and most, I think, the challenge that I will spend most slides on. We should search to understand the evolution of euglenophytes. We should search for missing links. And we are actually quite lucky that thanks to the work of Yamaguchi et al., we already have one, which is Rapaza viridis, a euglenophite that sits on exactly the position between heterotrophic and autotrophic euglenids that eats green algae and has plastids of who know what origin. And this Rapaza should definitely be studied more in the future, and this will bring a lot of knowledge about how the euglenophytes evolved. But what I would like to stress that we should also hunt other missing links outside, because there is a lot of environmental data available from Tara Oceans and Ocean Sampling Day campaign, for example, and we have peeked already into it. And in these stations that are in color, we found various euglenids data. And if we analyze the 18S and 16S ribosome RNA gene diversity, we indeed found quite a large diversity between between phototrophic and heterotrophic euglenids. As you can see here, there is a single known rapaza, but a whole bunch of other reeds from forming quite new clades, which would be interesting to get into the lab. And the same applies for 16S, in which we found a whole bunch of reeds locating somewhere between euglenophytes and green algal part of the tree. And I would like to stress also the work that I illustrate here by, I believe, the latest article of Gordon Lux and Alastair Simpson, who do very nice work on heterotrophic euglenids, which are depicted beautifully here. And this is the latest scheme of the understanding of the phylogeny. Not all relationships are understood. This needs to be improved. And to understand the origin of the chloroplast, we should specifically focus on these heterotrophs that are close to the point where the endosymbiosis happened. And I will close our contribution by the sixth challenge, which has been, it's a variation of several notes that have been already said, that we need genomes of other euglenids, and not only of heterotrophic euglenids, and not only good genomes, well, definitely we need good genomes, but at least some genomes, but also of heterotrophs. And by this, I would like to say that in our lab, we also try to sequence the genome of this heterotroph Rhamnomonas costata, which I already mentioned. Unfortunately, the quality of the assembly is not so good is nothing to be proud of, basically. However, we can still, it's not worse than Euglena gracilis genome, by the way. And it's again, quite fragmented, and it's derived from single-cell amplified DNA, which also doesn't help. But it helps us to, for example, to show that, or estimate that the genome size of this heterotroph, the haplogenome size, is smaller than Euglena gracilis, so the heterotrophs might be a good candidate for an easier candidate for genome sequencing. The transcriptome that we sequenced proved to be quite useful, and we learned quite a lot about the biology of this organism. For example, about the mitochondrial metabolism. And so I'm looking forward to similar studies like this, not only on phototrophs, but also on heterotrophic euglenids. And now I would like to summarize the challenges that we put together with Michael. And so we think we should determine the effect of a photostrophic lifestyle on the nuclear and genome and mitochondrial genome, and pay attention to the genome mosaicism to understand where the genes of euglenids, and not only of Euglena gracilis, came from. We would like to also, we should understand the biological meaning of the plastic genome structure and intron evolution. Are the introns all in junk, or do they have any function? For example, a regulatory function for the gene expression, and why they are so abundant in some. What's the role of inverted repeats or tandem repeats of which we see in the plastic genomes? The Professor Schwarzbach enigmas I've already mentioned. Why 3 membranes, not 4 as we expected? Did the plastid originated by mesocytosis and had the 3 membranes from the very beginning, or was one membrane reduced, and which? How the proteins transport across the membranes, particularly the middle membrane, which is still quite mysterious. I would stress that we need to investigate Rapaza viridis. This is I think, very good organism and very good missing link, and our field is actually lucky to have this. So we should study it and establish the values of phylogeny of euglenids, including the heterotrophs, to know who were actually the hosts of the endosymbiosis, and obtain well-assembled genomes in wide spectrum, from wide spectrum of lineages of euglenophytes, and not only genome but also transcriptomes, will of course help, as many other speakers mentioned already. And I would like to thank Michael for collaboration and you for attention.
Okay, thanks both. Pretty much bang on time. Thank you, Vlada. So comments, questions. Could I kick off with a very quick question? So with your assembly of the castata, were you running, do you think you were running into the same obvious problem that we had with Euglena, that essentially was a huge amount of repeat that just basically got dumped because we couldn't assemble it?
Well, I was not doing the assembly myself. I need to say that we started from whole genome amplified DNA because the culture is heavily contaminated by prokaryotes, as you can see. So we picked the cells, then we amplified the genomic DNA. And so from the very beginning, during the amplification of DNA fragments, so from the very beginning, we had fragments. And then we used only Illumina. We didn't even try to use any long reads because it would make no sense with short fragments to make a long read. And then, yeah, so I haven't done it. It was, the assembly was done in spades. And I actually don't know how much reads were lost, but yeah, it was difficult.
So the stats do look horribly similar to what we had for Gracie. So just cursory look at it. Okay, so questions. Susie Arnott says junk or functional. Maybe she could pitch in and expand on that comment.
Yeah. So sorry, I don't see the question.
It's in the chat. She says, Susie Arnott says junk or functional. That's the big question. My screen is like the third or fourth one up.
So I think it relates to the introns in the genome, in the plastic genome, right? Yeah. Well, I think it's just open question. I know that there has been some studies showing that the introns in the plastic genomes of some algae may have regulatory function because their splicing is affected, for example, by the temperature, like the rate of splicing is affected by the temperature or some other factors. So in theory, we can imagine that, well, this could also be involved, and there could be a situation like that. On the other hand, yeah, maybe the expansion of these introns is just because the environment of Euglena, where these introns get from the green algae, just didn't keep them under control. I don't know, that's what I can think of.
Very much an open question at the moment. So from Ross Lowe, how much of the observed Mosaicism is due to database bias?
Well, that's a good question. And I think, well, to understand this, we would need to do several experiments with various database contents and compare the results, I guess. And so definitely in the data that I've showed, the database was quite focused on algae and algal groups and green algae specifically. For this reason, we might see increased green algal content in the Rhabdomonas costata, for example. But we did actually this experiment in which we lowered red and green algal groups to the same amount. And if I remember well, we still did increase green algal content in euglenophytes, and I don't know what was the situation in the Rhabdomonas costata. So definitely the database content affects the results of these studies.
Yeah, totally. I fully concur with that some of this is quite possibly noise, that there is a cutoff there, that some of these things that are being predicted are probably slightly not robust predictions, I guess is the right word. Okay, so from Ellis O'Neill, do we know anything about the contaminants? So I assume he's referring to the Rhabdomonas friends.
Yeah. Okay, so we know that the Rhabdomonas data are contaminated by prokaryotes. For example, we have found there basically a whole genome of Tolomonas bacterium, which we easily removed. But still, when we are analyzing the data, we often see genes or proteins almost identical to bacterial proteins, so which we are removing. But the decontamination, it's kind of, you never have it 100%. So we know they are contaminants.
Yeah, almost impossible to clean things. So the comment from Alistair Simpson, he says he has a comment about phagotrophic euglenids. So Alistair, could you?
Yeah, can you hear me?
Yes.
Yeah, so thanks to Vladimir in particular, and also to Eric for showcasing the need for information on phagotrophic euglenids. Because I think from a large number of the questions that both of them brought up, both of the last two teams brought up, you'll need comparative information from phagotrophs. One thing that's important for context is that large tree of dominated by phagotrophs that Vlada showed was based on our work, which is overwhelmingly done using single cell methods rather than from cultures. So the availability of cultures across the phagotrophic part of the Euglena tree is very spotty. And for example, I'm not aware of any cultures, beg your pardon, from the phagotrophic lineage that is the best estimate for the sister group to green euglenids. Other groups are well covered. So the best estimate for the deepest branch within euglenids, there are several cultures available, but there are some big holes in terms of culture availability across that presently across that phagotrophic portion of the euglenoid tree. So that's really a plea to, when you're thinking about these missing links or filling in diversity, as Vlad was talking about, there's an important aspect in the phagotrophic part of the tree for that as well, especially if you want cultures. And then also all those phagotrophic cultures that we have, as far as I'm aware, every single one of them, with one possible exception, is a culture with one or more prokaryotes, and often with another eukaryote as well. So we're not talking azenic cultures there. Yeah. Cheers.
Okay. Thank you very much for that. Yes, that is very important. And so Scott Farrow has also asked, are these contaminants or symbionts?
So definitely, if speaking about Rhabdomonas costata, which by the way is not phagotroph, it's osmotroph from this Aphagia group that I'm pointing at. So it definitely does not have endosymbionts, or I think it doesn't have endosymbionts because we don't see in electron microscopy any bacteria inside. On the surface, attached to the surface, we also don't see bacteria. So definitely not like connected symbionts, but yeah, it could be that in the culture there is some symbiotic relationship based on some metabolites, for example, but this we cannot grasp.
Yeah, absolutely. I mean, it's obvious that most of these are extracellular as well, so we're not dealing with an endosymbiont. We're dealing with something that's presumably, if it was, connecting to the pellicle in some manner. Okay, are there any more questions or comments? Anything for Michael concerning comparative mitochondrial studies?
I think Alistair has a comment. Alistair Simpson.
That's already dealt with.
Okay.
Yeah, that's done with. Okay, so I think if there are no more burning issues, obviously the chat will stay open, and I guess we can take a coffee break or comfort break or whatever. And can I ask that we come back as scheduled at 5 minutes to the hour?
Okay.
So
thank you.
Everybody in the session so far.
I think we will try with Michael to answer the questions on the chat which appeared during the
Okay, very good.
time.
Okay. That's great. Excellent. So, yeah, so I think we have just a 10-minute coffee break. So, thank you to everybody, all the speakers so far, and to everybody who's put through some really interesting questions. And yeah, see you soon.